Hey everyone, and welcome. Have you ever wondered if you could, unmix colors? It kind of sounds like magic, right?
So, let's start with a simple question. Think about a regular black marker. You draw a line, it looks black. Solid. Simple. One color. But is that really the whole story? Is it actually just black?
You're not going to believe this, but no, not at all. That single, boring black line is secretly a whole mixture of different colors. It's like a hidden rainbow just waiting to be revealed. So let's go ahead and pull back the curtain on this little magic trick. We're going to explore the secret colors that are hiding inside the ink you use every day.
And we're not just going to talk about it. Oh no, we're going to prove it. And we're going to use a surprisingly simple scientific technique that, honestly, anyone can try at home.
The hero of our story today, the key that unlocks all these hidden colors, is a technique called chromatography. Now, I know it sounds a little complex, but stick with me. So what is it really? Well, put simply, chromatography is just a way to separate all the different bits and pieces in a mixture. It lets you take something like black ink and actually see every single individual color that was mixed together to make it. So how does it work? Well, chromatography needs two main players. First, you've got what's called the stationary phase. That's just the thing that stays still, like our paper strip. Think of it as the racetrack. And then you have the mobile phase. This is the liquid that does all the moving, like water or alcohol. This is our traveler. The whole separation happens when the traveler moves up the racetrack, carrying the ink with it. And here's the absolute best part. You do not need a fancy, high-tech lab to see this in action. Let's walk through exactly how you can set up your very own chromatography experiment right now. You probably have most of this stuff just lying around the house. Some filter paper, or hey, even a paper towel works great. A pencil, a jar, some water, and of course, the markers you want to test. See? It's super accessible.
Okay, step 1. Get your paper ready. Cut a strip that'll fit nicely inside your jar. Now here's a really important tip. Use a pencil to draw your starting line. Why a pencil? Because the graphite won't dissolve and run up the paper like pen ink would. It stays put. Then, just pop a small, concentrated dot of your marker ink right in the middle of that pencil line.
Next up, the jar. You just need a tiny bit of solvent in the bottom. And listen up, because this is probably the most important rule of the whole experiment. The liquid level must be below your pencil line. You don't want to dunk the ink dot directly in the water. You want the solvent to climb up the paper to get to it. So just hang the paper so the very edge of it just barely touches the liquid.
And now for the really fun part. Cover the jar. That just helps keep the solvent from evaporating, and just watch. Seriously, that's it. As the solvent starts to creep up the paper, you'll see the colors in the ink could begin to separate and race towards the top, all at different speeds. It's so cool.
So once the solvent gets near the top of the paper, go ahead and take it out and let it dry. Now, what in the world are we actually looking at? Let's figure that out. This beautiful strip of separated colors that you just made, it has an official scientific name. It's called a chromatogram. So congratulations, you're officially a scientist who has just created a chromatogram. So why did the colors split up like that? Well, it all comes down to a race. The colors that travel the farthest up the paper are the ones that dissolve really well in the solvent. They're happy to go along for the ride. The colors that barely move and stay lower down are the ones that stick more to the paper itself. So it's basically a competition between how much a color likes to dissolve versus how much it likes to stick. Okay, seeing the colors separate is awesome. But, in science, we love to measure things. So how can we turn this really cool visual observation into some actual comparable data? We can do that by calculating something called the retention factor, or for short, the RF value. The formula looks a little sciencey, but it's super simple. You just divide the distance your colored spot traveled by the distance the solvent traveled. That's it. This gives you a unique number for each color in that specific solvent. Let me give you a quick example. Let's say your solvent traveled 8 centimeters up the paper, and a little blue spot traveled 4 centimeters. You just do the math. 4 divided by 8. That gives you an RF value of 0.50 for that blue dye. Simple, right? It's a really powerful way to identify different substances. All right, before you run off to start unmixing every color in your house, let me give you a few pro tips and some other fun ideas you can try. Here are a few common mistakes to watch out for. Remember, keep that ink dot above the waterline. Use a really small, dark dot for the best, sharpest results. And always keep that lid on. Oh, and if nothing happens, don't give up. Some markers are permanent, which means they won't dissolve in water. Try a different solvent, like rubbing alcohol, and see what happens then.
And please, don't just stop at markers. This technique works on so many different things. Try crushing up a spinach leaf, get some of that green juice, and dab it on the paper. You might be totally shocked to discover hidden yellow and orange colors that are in the leaf the whole time, just hiding behind all that green chlorophyll.
Chromatography shows us that there's this incredible hidden world of color and complexity all around us, in leaves, in food coloring, in the pens on our desks.